Greetings, biology students. Today I'm going to teach you a little bit about genetic technologies. Like I've been saying for several weeks now, the field of genetics is growing a lot recently, and we're discovering brand new genetic technologies every single day. And I'm going to let you in on some of the more interesting ones that are widely used all over the place. These range from very old genetics technologies. The very first one I'm going to teach you about is pedigrees, and they've been used for at least hundreds of years. And then we're going to get all the way into like cutting edge advanced stuff like DNA microarrays. But let's start with just understanding what genetic technologies are. When I talk about genetic technologies, I'm talking about tools that scientists use to analyze and in some cases change DNA. So these technologies in some cases just allow us to read DNA, because remember that DNA is very, very, very tiny, so small you can't even see it. So just the part about reading DNA takes some very interesting and unique and rather challenging to develop tools, even to be able to read the code at all. And then we're getting to the point now with our technology that not only can we read that teeny tiny itty bitty little code that's inside every one of your cells, we're actually getting to the point now where we can change bits and pieces of it to do things like fix genetic diseases or grow more crops on the same amount of land, or make plants and animals and livestock that have features that we want them to have. So let's start with a little list here of the types of technologies that I'm going to expect you to know about. First, we're going to talk about pedigrees today. That's a very old genetics tool that's been used for a long, long time to look at the history of traits throughout a family line. We're going to talk about restriction enzymes. That's how we actually go in and slice pieces of DNA at very specific regions. So restriction enzymes allow us to cut up DNA at specific codes. Gel electrophoresis is one way to analyze DNA. You've seen gel electrophoresis done if you watch like CSI shows and stuff. They're always looking at somebody's DNA fingerprint. Well, the way you make somebody's DNA fingerprint is by running this tool called a gel electrophoresis. And every individual would have a unique readout when you do a gel electrophoresis. So that's how DNA actually gets read. Crime scene investigators and stuff use it all the time. But so do research biologists that want to just learn about the genetic code. Use a lot of gel electrophoresis. Recombinant DNA, that's making creatures that have inserted DNA in them. If you know somebody that's diabetic, they probably take insulin shots. Those were made by recombinant bacteria. We've had them for a long time. That's how insulin gets made still right now to this day. So that's putting genes from one organism into another organism. We'll talk a bit about that. Genetic cloning. It's not what most people think. Most people hear cloning and they think of like science fiction movies and stuff, and that's not really what we're talking about with gene cloning. It's definitely an interesting topic. It's complex enough that I don't want to get into it right now. PCR, polymerase chain reaction, is a unique way where we can take a little bit of DNA and make several billion copies of it. And then DNA microarrays are the gene chip technology that I keep going on and on about because I find it very exciting. This type of technology is going to give the general public access to their own genetic code. So well within your lifetime, probably well within the next 5 years, you will know your own genetic makeup. So you'll understand what types of diseases or disorders you're likely to get, and which ones you're likely to pass on to your offspring. So let's start with pedigrees. And we're going to look at those in detail today. Pedigrees, what you're probably familiar with is the dog food. But we're not talking about that kind of pedigree. Well, actually, we kind of are. The reason that pedigree dog food is called pedigree dog food, let's bring pedigree dog food back up here. The reason that pedigree dog food gets called pedigree dog food is because people that breed animals, like dog breeders and stuff, that breed dogs to show them, they worry a lot about the genetics of those dogs. And so in dog breeding circles and stuff, when people would breed dogs for like show dogs and stuff, they worried so much about the genetics of the dogs that they'd hand around these things called papers. Like dogs, if you buy a nice show dog, it comes with papers. The papers that dog breeders and horse breeders and stuff are always talking about are pedigree charts. And some dog food company figured out that all these breeder people were always talking about pedigree, so they named their dog food Pedigree. But that's where the dog food name came from. Pedigrees, what they really are, it's a tool used to figure out the history of a genetic trait in a family. And what else do we need to know about pedigrees? There's 5 basic rules to follow. If you go and you buy a racehorse, or you buy a show dog, or you buy some corn seed for your farm, it'll come with papers, and these papers will have these pedigree charts, and on these pedigree charts there's just 5 basic rules you need to know about. First thing you need to know, on a pedigree chart, boys are represented with a square, and girls are represented with a circle. And there's an easy and sort of sexist way to remember that, but it'll stick in your head forever if I tell it to you this way. Boys are big and boxy, and girls have curves, which I know that's kind of gross, but that'll help it stick in your head forever and ever. On a pedigree chart, you see a circle, it's a girl. You see a square, it's a boy. If it's filled in, it means the genotype is homozygous recessive. Remember that I told you need to get good at Punnett squares because they're not going away? Well, look, they're not going away. We're still going to use the same Punnett square terminology that we used before. You're going to need to know it again. So if you see a filled-in square or filled-in circle, it means the genotype of that person for that gene is homozygous recessive. If it's not filled in, it's homozygous dominant. If it's halfway filled in, it's heterozygous, which makes a lot of sense. It has one big version of the gene and one little version, one dominant allele and one recessive allele. Now, you need to know that for every single gene that we're going to look at, you're going to have a separate pedigree chart. So if you're looking at the family history of eye color, you'd have one pedigree chart. And then if you're looking at the same individual, but you're looking at the gene of height, you'd have to have a separate pedigree chart for the height genes than you would for the eye color genes. So that's why these, like show dogs and racehorses and stuff, when they come with papers, they'll come with just books and books and books of papers because you want to trace the family history of virtually every gene in that animal. And so they just have hundreds and hundreds of pedigree charts that come with that one animal. It's kind of part of the trick to why breeders and stuff make so much money doing professional dog breeding and horse breeding and livestock breeding. All right, I digress. A horizontal line directly connecting a square and a circle means that mating has taken place. So I'll show you what I mean by that in a minute because we're going to draw some pedigrees here in just a second. But if it's a horizontal line that directly connects a square and a circle, it means they mated. A vertical line separates generations. So the parents separating the parents and the babies will be a vertical line. And then finally there's one more type of line between your squares and circles. If it's not directly connecting the squares and circles, it means that they're related as siblings. So the lines and circles that drop in from the top, not connected side to side, that means that they're related as siblings, but they're not mating. So I'll draw some pictures and show you what I mean here in just a moment.